Hello, everyone. This is my AP Biology review video on Chapter 18, Regulation of Gene Expression. So the main things I'm going to be covering are basically all of the different ways that a particular gene can be more heavily expressed or repressed, and everything in between. So how is a gene expressed? How does it affect your phenotype? What are all the different mechanisms by which that gene is expressed and controlled, right? All that stuff. So to start off, just some general vocabulary that's going to show up throughout the rest of this unit. So anytime you even talk about gene expression, you're probably going to need to know these vocab terms. So first of all, you have an operon, which is just an entire section of DNA that is controlled by a single promoter. So for example, you have the lac operon, which involves the transport of lactose in prokaryotic organisms across the plasma membrane. So this entire operon is controlled by a single promoter. So if a repressor binds to that promoter, then that gene is no longer going to be expressed, and that lactose is no longer going to be transferred across the membrane. So next up, you have what's called a repressor, and a repressor is pretty much just a protein that directly or indirectly prevents a ligand or enzyme from binding to a target section of DNA. And what I mean by directly or indirectly is what we learned about in the enzymes unit a while back, which there's competitive inhibition, in which that protein just takes up the spot where the ligand/enzyme was supposed to bind. Or you have allosteric inhibition, in which the protein goes through the back door, I guess, and it basically changes the shape of the entire protein, preventing the ligand or enzyme from it just prevents it from binding to that initial site, just because the shape of it has changed now. But when the repressor binds, it's going to also prevent the expression of the gene. So then the promoter is pretty much where proteins and enzymes usually bind in order to start the expression of a gene. So maybe a process such as transcription. So the initiation step in RNA transcription, which we talked about in the last video, where you have all the transcription factors and then the transcription initiation complex. So all that happens right at the promoter, but there could also be a repressor that binds in the promoter region to prevent that initiation complex from actually doing anything. So this is where all the action happens, is the best way to put it. And then you have something called a co-repressor. And a co-repressor is a molecule that binds to the repressor and indirectly regulates gene expression. And what I mean by indirectly is that the co-repressor binds to the repressor, and the repressor is going to prevent the expression of the gene. And this could be, co-repressors are often found in feedback loops. So maybe a molecule that's produced by a certain enzyme that is coded by a certain gene. So that molecule that's produced at the end of the process is going to bind to a repressor, which prevents that same molecule from being produced more. So basically, the production of the molecule is going to repress the further production of that molecule. And then you just have this image up here, which is going to show some of the basic vocab, and it's a bit of a review from last time as well. So you just have this promoter right here, where the RNA polymerase II binds in order to start transcription, along with the transcription factors and the entire complex. And then down here, you just have the intermediate RNA processing step. So the spliceosome splices out the intron, binds together the exons, adds a GTP cap, adds a poly A tail for stability and prevents degradation of the RNA molecule. All that stuff is just a review from last unit, really, but yeah, that's helpful. So then let's just talk about regulatory sequences and regulatory proteins, which are pretty vague terms that describe many different things. But in general, regulatory sequences are sequences of nucleic acids capable of increasing or decreasing expression of genes. So this is literally a section on nucleic acid that can influence the expression of nearby genes. So then you have regulatory, and I think that's pretty self-explanatory, so I'm not going to elaborate too much. You have regulatory proteins, and these are proteins capable of increasing or decreasing expression of genes, especially prevalent during transcription. So a good example of this is just a repressor protein, which is a protein that repressor can bind to the promoter, and that can decrease the expression of genes. But then you could also have a molecule that increases the expression of genes, which there's another term for that, I believe, but I don't remember that at the moment. So regulatory sequences can significantly impact the phenotype of an organism based on which genes are impacted by those regulatory sequences. So for example, if a gene that determines eye color is expressed more because of the impact of regulatory sequences, maybe that eye color gets magnified in color, or the color changes entirely. You don't really know based on such a broad example, but it could significantly change the phenotype based on how much it increases or decreases the expression of that gene. So then you come to epigenetic modification, which sounds complicated, and it is a bit complicated, actually, you're right. So first off, you have something called the DNA methylation. And before I get to that, though, epigenetic modification literally just means a change above the DNA, which is what epi means. And then genetic means information, so change above the information. So as that name implies, the methylation and acetylation just add either an acetyl group or a methyl group right above a section of DNA. And because you added that molecule right above the DNA, it is going to change the expression of the gene. So for example, you have DNA methylation, which is the addition of a methyl group above a section of DNA. And this is going to lead to tight coiling of that section of DNA, which is going to prevent expression. And this is going to, so this is going to lead to the repression of the gene for the most part. So a really good example of this are the neurons in your brain. So these are heavily methylated because these are lifelong cells that are never actually going to unwind and be expressed. They're there, but a lot of them are going to be heavily methylated just to protect from outside threats, whatnot, right? And then I guess just another example that you have skin cells, which they're not going to be heavily methylated because they're super short-lived. They're super short-lived and they die very quickly, and they need to be expressed rapidly in order to achieve the purpose. Maybe that was a bit confusing, but trying to get a point across there. And then the other epigenetic modification is histone acetylation, which is going to be the addition of an acetyl group to the end of a lysine amino acid. And this is going to cause an increase in the expression of nearby genes. And this image down here is showing an example of methylation. So you have cytosine here, and you add this CH3 methyl group, and that is going to prevent, or that's going to coil up that gene and heavily suppress the expression of it. And then you have some more general stuff. So we have cell differentiation. This is a really important concept, actually. So you have a bunch of different types of specialized cells within your body. So like muscle cells, brain cells, nerve cells, skin cells, a whole other, like a million, probably thousands, maybe even hundreds of thousands, maybe even millions of different other types of cells. And all of these cells are going to have different functions despite having the same genome in them. So how is it possible that despite having the same genetic code, they all do different things? Well, this is due to differential gene expression, which is the expression of some specific genes within the entire genome. So most cells are not going to express the entire genome and every gene within it. They're going to pick and choose which gene they express based on their function. Excuse me. So the location of regulatory sequences along the DNA heavily influences their function, as their location is going to dictate which genes and functions of those genes the sequences impact. So if it's near a gene that codes for eye color, then the function that relates to eye color is going to change. If it's related to the immune system and the defense against foreign threats, then those regulatory sequences are going to play a big role in changing the function of the immune response. So then another important concept is the different types of operons. So the entire thing that can be controlled by a single promoter. So firstly, you have an inducible operon, and these are operons that are usually off. So they're not being expressed, but they can be turned on by a molecule called the inducer. So an example is the lac operon, which is going to be off unless a bacterial cell needs to transport lactose across the plasma membrane. And this is pretty easy to remember because inducible literally means you need to turn it on in order to do something. And the other type of operon is a repressible operon, which is an operon that is usually on, but it can be turned off. So an example is the TRP operon, which codes for the amino acid tryptophan in prokaryotes largely, I believe. And this is very important for bacterial growth and development. So it usually needs to be on most of the time in order for the bacteria to stay living, because tryptophan is a very important amino acid for that particular organism or prokaryotic organism. So it shouldn't be turned off because it's necessary for survival. Finally, you have miRNAs and siRNAs, which is quite an obscure topic. We don't really talk about this much at all, but just in case you need to know it, here's what you need to know. So miRNAs are microRNA, and these are very, very small sequences of RNA nucleotides that are cleaved from larger RNA. So by very small, I mean like 20 to 30 base pairs usually. Could be larger, could be smaller, but it's around that range a lot of the times. So miRNAs or microRNAs play a large role in RNA interference by destroying microRNAs made by specific, or sorry, not microRNAs, messenger RNAs made by specific genes to suppress gene expression. So to make that a little bit easier to understand, the microRNAs can often destroy, or at least significantly destroy, the messenger RNAs that are coded for by certain genes. And since the messenger RNAs are the things that are eventually going to create the proteins, by destroying the messenger RNA, you're going to suppress the expression of that eventual protein and the expression of a gene. Then you have an siRNA, which is silencing RNAs. And this is a double-stranded RNA molecule, which is very surprising because RNA is usually single-stranded. I have no clue how it works as a double-stranded RNA molecule, but that is what a lot of information is telling me. And yeah, this is also very small, 20 to 30 base pairs usually. These silencer RNAs contribute to the silencing of an entire gene, just its namesake, which is going to effectively stop them from being expressed. So yeah, that is the entire presentation. And with this video, that officially concludes the entirety of Unit 6. So all of the sickle cell unit is officially done. This is in 3 separate videos, since if it was combined, it would be like 45 minutes. That's too long for one sitting probably. But it's just to recap everything that we've covered this unit. We have DNA replication, all of the enzymes involved, some other basic concepts such as semi-conservative DNA strands, differences between eukaryotes and prokaryotes, what a plasmid is, the entire process of protein synthesis, mutations, RNA processing, and then all of the stuff you need to know about gene expression. So epigenetic modifications, miRNAs and siRNAs, transcription factors, inducible and repressible operons, there's probably some others that I'm forgetting, but that's a lot of what we covered. So hopefully these videos were helpful in helping you understand what I consider to be one of the harder units in AP biology. So thanks for tuning in. And I will see you next in the heredity units, which is the one I struggled with a lot. So it'll be helpful for a lot of people.